So now we are moving to the examples of culture finding. So we'll see various images here. So again, like it's going to be the cleft agar. It's going to be the cysteine, lactose, electrolyte deficient medium. So here there's going to be a deficiency of electrolyte, and there's going to be a cysteine, lactose. So here like you'll be able to determine whether it's going to be lactose fermenting or non-lactose fermenting. So here like you are able to see more than 10 to the power 5 colony forming units per ml lactose fermenting colonies. So this yellow color colony, and there's going to be a pinkish hue, it's going to be a lactose fermenting colony. Example is going to be the Klebsiella species, and we have the Escherichia coli, which is going to be lactose fermenting organism. And we have the second, that is the cleft agar, which shows more than 10 to the 5 colony forming units per ml of urine of non-lactose So usually we will regard them in the laboratory as LF and NLF. In case it is going to be a lactose fermenter, we tell it as LF, and in case it is going to be a non-lactose fermenter, we tell it as non-lactose fermenting colony, NLF. So example for non-lactose fermenting organism, Pseudomonas, and we have the Proteus species like Proteus mirabilis, Proteus penneri, and Proteus vulgaris, and other associated organisms like Morganella morgani. So it's going to be a non-lactose fermenting organism. Now we are going to the third. So again, like you are able to see cleft agar, we are able to see more than 10 to the 5 colony forming units per ml of, is going to be an lactose fermenting or non-lactose fermenting. Yes guys, are able to answer this particular question? So very good, like it's going to be a mucoid lactose fermenting colony. So example for mucoid lactose fermenting colony, it's going to be Klebsiella species. And again, it's going to be an more than 10 to the power 5 colony forming units per ml of white. Yes guys, so here like you are able to see what type of colony. So if you are able to see closely, like you will be able to see the growth of white creamy pasty colonies. So again, like it's going to be a non-lactose fermenter. So the example is going to be the Candida species. So again, like whenever like there is going to be a growth of any organism in the culture plate, so what we are going to do is that, sir, we are going to do the gram stain, and we are going to do the biochemical appropriate for that particular gram stained picture. And then with that, like we can do the antibiotic susceptibility testing. So this is the final image. So here like you are able to see the fourth slide, that is the cleft agar, like non-lactose fermenting creamy pasty colony. So that's going to be the Candida species. So we have the fifth one. So here like it's going to be again an cleft agar, and here like you are able to see more than 3 types of colony. So this is one type, and we have this is one type, and this is one type, and we also have yellow colored colony, and we also have an white color colony. So here like it is going to be more than 3 types of colony. And it does not quantify the particular cause concept, or it will not satisfy the cause concept. So it's going to be the reported as mixture. So again, like we are going to test the particular sample with the repeat sample. We are going to collect the repeat sample and we are going to do the testing procedure. So mixture. So it is due to the failure to obtain clean catch midstream urine sample. So whenever there is going to be an urine analysis for culture sensitivity, like we need to obtain a specimen called CCMSU, which stands for clean catch midstream urine sample. So here the patient must avoid the front part and the final part of the urine, and they are supposed to catch only the middle part of the urine in a sterile container and going to submit it to the laboratory for culture and sensitivity testing. So here, like it's going to be more than 3 types, so it's going to be a mixture. So this kind of particular culture plate, we are not supposed to perform any antibiotic sensitivity testing or any identification. So here is going to be more than 3 organisms, so we doesn't know which is going to be a pathogen here, and it's going to be an beyond the significance. So it will not tell about the significance of the pathogen which is involved in the infection.